We have one more category of growth promoting onc genes to talk about, genes that code for cyclins or CDKs. So let's look at this, and it's a very important category. CDKs phosphorylate target proteins are expressed constitutively, meaning that CDKs, cyclin dependent kinases, are always present in the cell. They are constitutively expressed. During the cell cycle, however, they are in inactive form. However, cyclins are synthesized during specific phase of the cell cycle. So as the name implies, cyclin-dependent kinases can only function, here is a CDK4, when cyclin D is present, is available. So this binding of cyclin D with CDK4 results in conformational change of CDK4 molecule, exposing the enzyme domain, enzymatic domain, the active site, which can then phosphorylate other proteins. One of the targets of cyclin D in association with CDK4 is RB protein. We have talked about previously about this. I briefly mentioned about RB. We are going to talk about RB in a lot more detail subsequently, but here I would just like to mention RB is basically a protein that binds DNA. It is basically a brake which is holding the progression of cell cycle. Once RB protein, RB by the way is abbreviation for retinoblastoma. It is in this disease this protein was first identified. So once this complex phosphorylates RB protein, the brake is released and cell can progress into the next phase of the cell cycle. In this case, it will be the S phase. So cyclins are, although CDKs are always present, cyclins are only expressed or made during specific phases of cell cycle. Activated CT4 and CDK both bind cyclin D, by the way. They phosphorylate RB, which overcomes the brake or the hurdle of cells going from G1 into S phase. Cyclins are also regulated by specific proteins which inhibit the function of cyclins. Here I'm going to highlight three important proteins. We are going to talk about these proteins later on. However, I would like to mention one of these proteins is P15 right here, P15, which is, I have marked P15 with the green arrow. P15 can inhibit the function of cyclin D and CDK4. The other important protein which I have marked with red arrows is P21. We will talk about P21 later on. So the red arrows are pointing towards P21. P21 can inhibit the cell cycle at different stages. As you can see, P21 can inhibit at this stage, and also at this stage. The other very important protein is P27, which is marked with a purple arrow. P27 we have already talked about. MYC, basically when it binds different sites on DNA, it results in transcription of proteins that will destroy P27, causing the cell cycle to progress. So I would just like you to keep this in mind, where we have seen these inhibitory molecules which inhibit the function of cyclins and CDKs. Cyclin D genes are overexpressed in many cancers. For example, the ones affecting breast, esophagus, liver, and subset of lymphomas. Mutations affecting other cyclin and CDKs also occur in some neoplasms. TGF-beta, here I would also like to mention there are certain signaling molecules that tell cell not to divide. TGF-beta, for example, is one of those molecules. It results in production of P15. P15 is right here. We have already talked about it. It results in production of P15, which inhibits the transcription of CDK2 and CDK4 and also cyclin A. So here we have just talked about how cyclins and CDKs, if there is a mutation in them, can result in neoplasms, and how there are other molecules that inhibit their function. We will continue our discussion of neoplastic transformations in next module.